On today's episode of Mind Matters, we're exploring the cellular mechanisms underlying the neurodegeneration and the innovative approaches to the intervention. So, mitochondrial dysfunction is very central to many neurodegenerative processes. Given that the brain's enormous energy demands, why do you think neurons are particularly vulnerable to bioenergetic failure? And could enhancing mitochondrial biogenesis or mitophagy be a universal therapeutic strategy across different neurodegenerative diseases, Nehal?
So yeah, neurons are especially sensitive to mitochondrial dysfunction because they require a lot of energy, which glycolysis alone cannot support. So boosting mitochondrial growth or clearing out damaged ones can help neurons stay healthy, but treatment needs to be customized for each disease and cell type to avoid causing new problems.
The concept of cellular reprogramming has shown promise in generating disease specific neurons from patient cells. How do you see patient derived organoids and induced pluripotent stem cells revolutionizing our ideals and our understanding of disease mechanisms? Are we moving towards truly personalized neurodegenerative disease models?
So, patient derived iPSCs and organoids let us study a person's unique disease by capturing their genetic and epigenetics. They reveal a lot about early disease mechanism, show patient to patient differences, and allow testing drugs on individual cells, but they are not perfect. They don't capture brain structure, effect of aging, and whole body effects, which are missing. So, combining them with bioengineering, microfluidics, and computational models is needed to fully understand the disease and create a personalized neurodegenerative disease models.
I agree. So another angle is of that of inflammation. So neuroinflammation walks a very fine line between protective immune responses and destructive chronic activation. So microglia can be both neuroprotective and neurotoxic, as we all know. So how do we develop therapies that preserve beneficial immune functions while preventing pathological inflammation? Is the answer in timing, location, or cellular specificity?
So, therapy should guide microglia towards protective role rather than completely shutting them down. So, the answer lies in timing because if we can catch the shift between turning off microglia from neuroprotective to neurotoxic, we can target specific region or cell program and prevent chronic inflammation while keeping essential immune functions intact.
Right. So now coming to autophagy dysfunction, which appears to be again very central to protein aggregation disorders. The interplay between ubiquitin-proteasome systems and autophagy lysosome pathways suggests a delicate cellular housekeeping balance. If we enhance these degradation pathways therapeutically, what are the potential unintended consequences of accelerated cellular turnover in the aging brain, do you think?
So, boosting autophagy and proteasome activity can clear toxic protein, but overdoing it may can harm cellular components, especially in aging neurons. So, the goal should be to create balance by controlled selective activation rather than maximum clearance.
Right. So, another angle of that is the synaptic dysfunction, which often precedes cell death in neurodegeneration. Given that synapses are dynamic structures constantly remodeling throughout life, how do we distinguish between pathological synaptic loss and normal aging related changes? Could synaptic enhancement be a preventive strategy even before symptoms appear?
See, normal aging slowly prunes synapses, while neurodegeneration causes it faster in circuit specific manner. So, biomarkers and imaging can tell us the difference between pathological synaptic loss and normal aging related changes. So, supporting synaptic plasticity early through lifestyle drugs and stimulation could help prevent symptoms, but it needs careful tuning to avoid overexcitation of neurons.
Yeah. That brings us to the end of the podcast, Mind Matters. For everyone who was listening, thank you, and thank you to you, Nehal, for being our host.
Normal aging slowly prunes synapses, while neurodegeneration causes it faster in circuit specific manner. So, biomarkers and imaging can tell us the difference between pathological synaptic loss and normal aging related changes. So, supporting synaptic plasticity early through lifestyle drugs and stimulation could help prevent symptoms, but it needs careful tuning to avoid overexcitation of neurons.
Yeah, that brings us to the end of the podcast, Mind Matters. For everyone who was listening, thank you, and thank you to you, Nehal, for being our
Guest.